Okay students, this is chapter number 2, diffusion and osmosis. In our previous lesson, we went through different definitions, diffusion, active transport and osmosis. We also see what's the importance of water potential gradient in the uptake of water by the plants. By the end of today's lesson, you would be able to tell the effect of osmosis on a plant cell and on an animal cell. Or you would be able to tell the effect of hypotonic and hypertonic solution on a plant cell and on an animal cell. We will also see the importance of Turgor in plants. To start our topic of effect of osmosis or effect of hypotonic and hypertonic solution on both animal and plant cell, let's understand this diagram first. If you see that I have taken 3 animal cells and 3 plant cells. This solution, which is labeled as isotonic solution, is such a solution in which the amount of water or the water potential is similar to that of the cell. Let's say there was 40% water inside the cell, 40% water is present outside the cell. Iso means same, tonic means the concentration of solute. So isotonic solution means when the concentration of solute is similar in both the sides, at both the sides, or you can say the water potential is same at both the ends. It is same in the cell as that it is in outside. It does not mean that water won't come inside or can leave it will do that, but in a way that the shape of the cell will be maintained because the water levels are similar on both the sides. Let's say 5 molecules of water came in, so 5 molecules of water would go out to keep the balance or to keep the cell in the shape. Same would happen with the plant cell. If you see that these cells are maintaining their shape when placed in isotonic solution. On the other hand, here if you see that I have taken an animal cell and I have placed in hypotonic solution. Hypo means low, tonic means solute concentration. So it means the solution in which this animal cell is placed has low solute and more solvent. Here we are considering water as solvent. So since water is greater outside compared to the cell inside, so water moves from higher water potential to low water potential into the cell through a partially permeable membrane, or you can just say that water moved into the cell by osmosis. It will keep moving into the cell, but if you see the cell is not maintaining its shape, in fact it swelled and ultimately burst. Animal cell is unable to stand itself in hypotonic solution because it does not have a cell wall to protect it. Cell membrane is very thin. When water would keep moving into the cell by osmosis, it will ultimately burst. Hypotonic solution will cause bursting of animal cell. But on the other hand, if you see, I have placed a plant cell here in the same solution. Water is moving into the cell because it is higher outside compared to inside of the cell, but the cell have become turgid. It did not burst. The reason is that it has cell wall which is flexible, tough, and it would maintain the shape of a plant cell and will prevent it from bursting. Now come to the third solution, which is called hypertonic solution. Hyper means high, tonic means solute concentration. So this is a kind of a solution where the concentration of solute is high and that of solvent or water is low. Low compared to animal cell, compared to plant cell. So when water is less outside compared to the cell, the water would move from the cell out of it into the solution by osmosis, shrinking the cell. This is called crenation. Crenation is the shrinkage of cytoplasm because of loss of water from an animal cell when placed in hypertonic solution. On the other hand, if you see plant cell, its cytoplasm and vacuole has decreased in size. The cell membrane here has gone with the cytoplasm. These double layered cell wall are staying on their place, but the size of the cell has been decreased because of the shrinkage of cytoplasm away from the cell wall. This is dark green shaded cell membrane, the two double layer of cell wall. These are the vacant spaces that have been created because of plant cell being placed in hypertonic solution. Water moved out of the cell by osmosis, shrinking and reducing the size of the cell, of the vacuole, of the cytoplasm. This condition is called plasmolysis and the cell is called plasmolyzed. Plasmolysis is the shrinkage of cell cytoplasm away from the cell wall so that the overall size of the plant cell decreases. It becomes plasmolyzed. Now this is the theory of what we have just seen in the diagrams. Effect of hypertonic solution on a plant cell. A plant cell will be plasmolyzed if placed in a concentrated solution or hypertonic solution. Hypertonic solution is a solution that contains a lot of solute and less solvent, or you can say it is kind of a concentrated solution. A concentrated solution to have high amount of solute and less amount of solvent. Water potential inside the cell will be higher than that in the solution. So water will move out of the cell by osmosis, vacuole decreases in size, plant cell will lose its turgor or turgidity and will wilt or will become limp or flaccid. All these expressions are correct and stand for similar meanings. Either write wilt, limp or flaccid, all of them are correct. Plasmolysis is the shrinkage of cytoplasm away from the cell wall when plant cell is placed in a hypertonic solution or solution of low water potential. What about animal cell if it is placed in hypertonic solution? It will lose water by osmosis because water was higher in the cell compared to the solution outside. The membrane of the cell would form spikes leading to crenation. Cell will be dehydrated and eventually die. What about hypotonic solution? Hypotonic solution is a solution which contains low amount of solute, or you can call this solution as dilute solution. The water potential of the cell sap is lower than that of solution outside the cell. Water will enter the cell by osmosis, vacuole will increase in size and will push the cell contents against the cell wall. The cell will become turgid. The pressure applied by water on the cell wall is called Turgor pressure. Plant cell will not burst because of the presence of cell wall. On the other hand, same thing will happen with animal cell. Water will enter into the cell when placed in hypotonic solution. It will swell, but unlike plant cell, it will burst. Reason being that it contains no cell wall. This is another diagram to help you understand the effect of hypotonic and hypertonic solution on an animal cell and on a plant cell. You see, I have taken some animal cells, RBCs, and I have placed them in distilled water and I have placed them in concentrated salt solution. Now distilled water is pure water or you can say it is hypotonic solution, or a solution with very low or negligible amount of solute and a lot of high water potential. What happened was that the normal shape of the cell did not maintain itself. The cells swelled because water entered into them, and ultimately they burst. On the other hand, if I will place them in concentrated salt solution or concentrated sugar solution or any other concentrated solution, water will be lost by these cells by osmosis. There will be spikes formed by the cell membrane of the cells. This is called crenation. Let's see about the plant cell. It was a normal plant cell. When placed in hypotonic solution, the size of the cell increased because of the entry of water into the cell. The vacuole size increased. The cell contents were pushed towards the cell wall. The water applied pressure on the cell wall. This is called Turgor pressure. But despite all the pressure applied on the cell wall, it did not burst and retained its shape, became turgid because of the presence of cell wall. On the other hand, if I place the same cell in hypertonic solution, the cytoplasm will shrink and will move away from the cell wall. The cell membrane will bound the cytoplasm. The size of the vacuole will decrease. This is called plasmolysis, and the cell is now called plasmolysed cell. There is a question in your past paper that what does these spaces contain? These spaces drawn or shaded with green color, what they will contain? Can you guess? If you can, let me know after the lesson. Okay, now we have to see importance of turgor in plants. What is the importance of turgor in plants? First point is that they maintain the shape of soft tissues in plants. Wherever the soft tissues are present, where there is no wood, turgor will provide support to these tissues which have no supporting structures. Turgor provides support to young stems and leaves of non-woody plants. I have shown you a non-woody plant here. If you see, they have soft stems and shoots. No wood is present for the support. In such plants, the support is provided by turgor. These plants are also called herbaceous plants or non-woody plants. They also help in the movement of certain plant parts. For example, in mimosa plant, this plant is also called as touch me not plant. Folding of leaflets is done because of changes in turgor pressure. If you see in this diagram, this plant is called mimosa plant or touch me not plant. Whenever it is being touched, the turgor is changed or lost by the leaflet, and the leaflet turns to fold itself. The other importance of turgor is opening and closing of stomata by change in turgor of guard cells. If you see in the diagram, the stomata opens and close. What controls the opening and closing of stomata is the changes in the turgor of guard cells. These guard cells are bean-shaped cells controlling the opening and closing of stomata. How they are closing or opening it? Whenever the guard cells are turgid, or you can say whenever the guard cells are full of water or are in their turgid form, the stomata is open. When guard cell lose water or when they lose their turgidity, the stomata closes. Other importance of turgor is opening and closing of flowers. Bending movements of petal is due to changes in turgidity of the cells on the opposite surface of petals. So if you see in the diagram, the petals are blooming, they are opening up. It is because of the fact that the cells on the lower sides have become turgid. Wherever the petals would be moving, the turgidity would increase on the opposite side. Like here they are opening in downward direction, so turgidity is increasing downward or in the cells which are present at the base.